So are we live? So, where do I have to stand? Anywhere? Okay, hello everybody. I'm Ellen Jorgensen. I'm the director of Genspace, the community biolab in Brooklyn. We're actually on the 7th floor, and after the talk, you're welcome to come up. I'll take people up for a tour of our messy little space afterwards if you'd like. We do a lot of different things. We act as a community resource. So the main thing is to provide access to modern biotechnology with a very low threshold. So we're part biohackerspace, meaning you can become a member for $100 a month and work on a project in that area. The only thing that we ask is that it conforms to biosafety level 1 guidelines, but we don't care if it's going to save the world or make money or even if it's going to work. It could be just an exploration that you're doing for your own amusement. And that's kind of an interesting concept because most other labs, professional biolabs, you have to be part of a university or company to access them, or you have to commit to paying for space for a year. Excuse me. And so Genspace has none of those restrictions. So we feel it's a very important space for people to discover this technology, to learn about it, and also potentially to invent new stuff. We have a very diverse group of people that works there, everything from artists and engineers and writers, hobbyists, professional scientists, lots of students. And we like to see that mix, even bio-entrepreneurs and VCs. So it's interesting to get that bunch of people cooking together. And that's what we like, is to foster this kind of interactive community. So this is part of our talk series, and we have talks at least once a month, and they're either artists working at the art-science interface or scientists who we have very strongly said to these people, we want you to explain your work in terms that the general public can understand. This is not like the usual lecture that you give in front of a bunch of your colleagues. This is your chance to tell people what you do for a living and have them see how awesome it is. And maybe they come away with a better understanding of your field and more sympathy for supporting that type of research. So it helps everybody all around. This is the first time that we've actually tried live streaming it. I hope this is working. Hello to everybody who may be watching. I'm gonna let our guest lecturer, Dr. John Borghi, who's a neuroscientist and works at Rockefeller University, have the floor in a minute. The only thing I will say is that this place is supported by donations and by memberships and by the classes that we teach. We don't have any external funding at the moment. So I'm actually going to scurry upstairs and put a little contribution jar, and if you want to put $5 in it, we would love that. It would help cover the food and the wine. So, all right, without further ado, I'm going to let John take it away.
So hi, everybody. Can everybody hear me in the back? No? Okay, I'll try to project as I talk. So my name is John Borghi, and I am a cognitive neuroscientist, amongst other things. So I got my PhD in neuroscience a couple years ago at Stony Brook University out on Long Island, and I did brain imaging research, so human brain imaging. So I put people in MRI scanners and had them do very boring things and then looked at their brain activity. And today I'm going to talk about kind of new and emerging neuroscience techniques and kind of the state of neuroscience as it is, because we're in a very exciting, very transitionary kind of period. And yeah, if you can't hear me at any point, or if you have questions, just let me know. I guess if you're watching online, you can't do that. But okay. So just, if you don't know, I just want to explain the title. I didn't get my cap lock stuck as I was typing this. If you don't know, there's a big neuroscience initiative President Obama announced 2 years ago now, the Brain Research Through Innovative Neurotechnologies initiative. So it's got its own name in the acronym, which is kind of weird. And it's to help develop it's a lot of money, a lot of partnerships between industry and federal agencies to develop new technologies to investigate neuroscience, amongst other things. Okay, so what's going on in neuroscience? There's a lot of large-scale kind of neuroscience initiatives happening right now, which is partly why it's such an exciting time. So I already mentioned the Brain Initiative. There's also, in Europe, the Human Brain Project, which was kind of conceptualized as the European version of the Brain Initiative. It hasn't turned out to be that, and I'll talk a little bit about that moving forward. And there's also Human Connectome Project, iWire, which is kind of crowdsourced neuroscience. People play a video game to help map neurons in the retina. And then there's things like the Open Connectome Project, the Open Worm Project, Blue Brain Project. We'll talk about mostly, if not all of these, moving forward. Okay, so I have a couple caveats before delving into technology. There's a thing that both pretty much worries me as a neuroscientist, and that is there's some research showing that people are more convinced by scientific arguments when they invoke neuroscience, even when neuroscience is completely inappropriate for the example. So basically, it's this idea that if you show a brain with like some colors on it, people will be convinced of what you're talking about, which is helpful for me as somebody who makes those pictures, right? But it's not good for our public understanding of neuroscience. So this was a study done in 2008 showing that if you make a circular argument and show a brain image next to it, people are much more likely to believe it than if you show just a bar graph or just the text. And I realize that I'm telling you this by showing you some brain images and a bar graph, but sorry. Yeah. Yes. Also, there's a lot of kind of neuroscience in name only or neuroscience loosely defined things kind of floating around right now. So you may have heard of neuromarketing, which is advertising agencies kind of invoking neuroscience technology in a way that neuroscientists may not find to be very good to try to sell companies their advertising services. So you might encounter this. This was a commercial that came out maybe a week or two ago where Porsche said that their new car will stimulate your prefrontal cortex, which is a completely ludicrous statement, right? Because even if it of course it does, it's a thing and you're interacting with it, but everything does that. They have a commercial, you can Google it, where they stick somebody with an EEG cap in a fighter jet and have them fly around in it, and then put them in a car and say, like, look, it's the same. Our cars are like fighter jets. There's so much wrong with that entire thing that I could talk about the whole hour. And there's also this kind of cottage industry of things that services people try to sell you. Cognitive enhancement or treatment for psychological illness or attention problems, invoking kind of neuroscience-y things. So we'll talk about a couple of these. Luminosity is like this huge industry. It's I think the brain game kind of industry makes more money than is given to neuroscientists in the US. So, so that's, we'll talk about some of that. And then, neuroscience being, yeah.
What about devices?
So the
interacts on that. It's raising a ton of money for Series A financing.
So these like wearable
Wearable,
yeah, devices. I'll talk about that a little bit, but the evidence for that, for their kind of efficacy in clinical, very well-controlled settings is mixed. And when you start just giving them to people who aren't trained and not working under these same precise conditions, they could cause really major problems if they cause anything. There's a subreddit on Reddit devoted to this kind of stuff, and it's like people listing like, oh, I tried this and I just burned my skull. I did this, you know. So don't try this at home. But we'll talk about that moving forward more. It's a big industry. There's a lot of like venture capital going into it right now, but I think that's a bubble that's going to burst because the actual science is not there at all. Okay, and then for whatever reason, things in neuroscience, maybe things in kind of biomedical stuff in general, tend to get really exaggerated in the popular press. So this was a study and a headline that came out a couple weeks ago where somebody, I think this was Time Magazine, said, being poor affects kids' brains, study finds, which, okay, sure, that's rather uninformative and kind of scary sounding. And then I looked into the actual study and they just said like, well, we did a correlational study, right? We looked at brain structure of people in very low socioeconomic status versus people in very high, and it looked like in our sample maybe there was some difference, but it's completely correlational. Obviously, we can take somebody's brain and make them poor and look at what happened. So you know, these things can get exaggerated a lot. So just, you know, be careful. Okay, so this is what we're talking about, right? The human brain. That's my brain, the one that's blue. That's my wife's brain, the one in red. I didn't force her to get a scan before we got married. We met in graduate school, and this was the research that I was doing while we were there. Okay, a couple more caveats. So this is a map of the brain made in 1909 by this guy Corbin Brodmann, and he sectioned a bunch of brains and did some cell staining. And I put this up here just to show that we as a field, are developing all these very amazing technologies, but we still use brain maps from 100 years ago. These areas are still, if you look at fMRI papers, they're still used to describe brain regions. And you know, we're in a kind of public lab space or community lab space here. And I'll be talking about things that cost millions of dollars, but this grid here, this is a Herman grid. And if you look at it, it might not work as well with the projected image, but you can, like, there's little dots in the, so this grid has been used, just this image to understand some neuroscience of the retina, because you see that illusion that was used to explain how some of the microcircuitry of the retina works. So, you know, you can still do neuroscience kind of on the cheap, right? Like, this is just a picture, you can put it up. And one more thing. So I'm sure everybody saw this picture here of this dress. There was a big meme going around maybe a month or so ago. Some people see it as blue and black. Some people see it as gold and something else. I don't know. I see it as blue and black, so I can't. Anyway, and then there are a couple of other illusions that came out after it that kind of capture people's attention. Is that cat going upstairs or downstairs? Is building 1 or building 2 in front of the other, right? So we can't really explain these things. These are very basic kind of visual things that our brain is doing, and we can't tell neuroscientifically why some people see that dress as different colors, why some people see that cat going up and down, or these things. So there's still a lot of progress that needs to be made on these very basic problems like color perception, depth perception, and kind of motion, right? Just today, so we're going to talk about cutting edge neuroscience. This is something that came out this afternoon. Somebody tried to investigate the dress, and basically it doesn't matter what these graphs are showing really, because what they determined at the end was, yes, some people see it as one set of colors, some people see it as another set of colors, which we knew, right? If I took a poll, we could have figured that out. Neuroscience, when this was all happening, I was getting some like calls and emails and stuff from people saying like, how is this happening? And all I could say was, well, our perception is not, it's not like a camera. It's affected by our thinking and our top-down kind of processing. But other than that, I don't know. Right? I have a PhD in neuroscience, and people who are much more decorated than I am were saying basically the same thing. So when we talk about neuroscience, we talk about a bunch of scary-looking diagrams, right? We're talking at multiple levels of looking at the brain. So I am a cognitive neuroscience scientist, and I work with human subjects. So I study kind of a macro level, like brain regions interacting with each other or activity in singular kind of big regions. I have a colleague from college who studies individual cells, right? And I was talking to him recently, and we figured out that we both say that we study the exact same thing, but at very different scales and in very different populations. He studies individual cells and looks at memory in a petri dish, basically. And I look at people walking around in the world, right? So that's very different. So I'm going to talk about some technologies, both of these levels, and we'll go from there. So this is a trace of one neuron and all the neurons connected to it. So you're getting a sense of how complex this can all be. This is called transsynaptic tracing. So somewhere in this mass, one cell was injected with a compound, and it crossed the synapse, and those are all the cells that are connected to that one cell. So that's kind of crazy, right? Like that's a hugely complicated wiring diagram, and that's one cell and one set of connections. And in the human brain, we have billions of cells that make trillions of connections. You can kind of get a feeling of how complicated this is. So what's kind of neat about this this technique is the compound that they use is a green fluorescent protein, which is used in a lot of biology, and a virus. And the virus that they used was rabies. So we're mapping the brain with rabies, basically. And what makes rabies perfect for this is it affects neurons and can cross the synapse. And this is a particular recombinant version of rabies that can only jump once. So it's pretty neat. There's other versions of this technique that use like herpes and different things. So very scary viruses that we've kind of taken control over to map the brain.
What's it by jumping once?
So one neuron down the line. So all these, so this dot is also connected to a whole bunch of things, but this technique only jumps to one. Once it does that, it can't go any further.
One week of this.
Yeah, exactly. I'm actually not sure. I don't think so. Kind of the problem with this is it doesn't, behave like a lot of things that neuroscientists are used to dealing with. As a neuroscientist, I don't often think about viral transmission from one cell to another. It's not a big thing. But I think it just hijacks the synapse kind of in general. So it depends on what you're injecting it into.
What is the red stuff?
The red stuff is just another trace. I think they were just highlighting this cell here. So this is a cool other version of, this isn't tracing neurons, but this is a form of microscopy that I think is really neat that just came out this year too. So this team, there's a lot of neuroscientists who work with microscopes, look at individual cells or look at things that have been sectioned, right? And the big problem is developing new microscopes. At a certain point with optical microscopes, you run into problems where you just can't make a better microscope. So this research group, I think they're at Harvard, decided to go the opposite route. Instead of making a better microscope, they just figured out a way to make their sample bigger. So by using a compound that's, what is it called? It's called sodium polyacrylate. It's a compound that's found in hyper water resistant diapers. Actually, they isolated it from diapers. They can make their samples bigger while maintaining kind of the shape and structure and structural relationships between everything. So they're literally not inventing a new microscope. They're inventing a new way to expand the samples. So this sample here, with the same microscope, you can see much finer detail. And they didn't adjust the microscope; they adjusted the sample itself. So that's pretty cool. And I don't know if anybody was reading the latest New Yorker. There's a profile of this guy called Desserot in it, and he's the inventor of two ginormously influential neuroscience techniques, Optogenetics, which we'll talk about in a minute, and this one called clarity. So this is a technique for making brains clear. Now, you might be saying, why is that? So what, right? Well, when you traditionally do neuroscience, not in humans, obviously, walking around the world like I did, but with animals or with plates or something, to look at the anatomy, you have to section things, right? You have to get in there and actually look at the sample. You have to take apart your brain. With this, you can get images like this, because these are all stained with a protein. But to get these images, you don't actually have to section. So you can look at things that are kind of all together, which is really, really neat. And by making the brain clear, and by kind of clearing away all this color, what you can do is you can improve your imaging for other kinds of techniques. So one such technique is called Brainbow. It's like rainbow, but with brains. Yeah. Neuroscientists are not super clever at that, I guess. So the traditional way of staining individual neurons to look at them was invented, in the 1800s, Golgi staining. So this is, some Nobel Prize-winning work from, 1907 or something like that, Ramon Cajal. And it's really cool, right? Like you can see neuron branches and you can see the cell bodies. The problem is it's all the same color, so you can't really tell which branches belong to which cells at a certain point. What this Brainbow does is it's a complicated genetic thing where they basically inject different animals with a recombinant version of these fluorescent proteins. And when they get into the cells, they combine in such a way that individual cells have different hues when under UV light. So you can see here that these different cells are different colors. So you can work disentangling some of this crazy mess here. Seeing these pictures makes me very glad I work with bigger things, because this is very scary looking. And when you combine something like this with clarity, you get very high resolution, very exciting looking images, right? Like you can see a lot of individual cells here. You can trace all their connections. So in terms of building models of the brain or looking at the microcircuitry of the brain, like this is kind of the new frontier. The problem is this is all super expensive and takes a very long time, but it's very cool. And this is another thing that was just announced today, actually. So again, cutting edge neuroscience. The Allen Brain Institute announced that it now has a database where people can look up individual cell types. And when you look under an entry, it gives you all kinds of information about that cell type. So that's very exciting for neuroscientists working at that level, because they can look at functional data, which is like how that brain behaves or how that cell behaves, as well as a bunch of other data, including morphology and things like that. So it's very exciting.
Can you explain some of the fields in that database?
Some of them. So dendrite, there's different dendrite classifications, basically. Spiny neuron is just one of them. Layered is which cortical layer it's in, so there's different layers in the cortex. This is obviously the morphology, and then there's some data about the timing, so how often it fires at a given time. So all cells, most neurons fire kind of continuously, it's just at different rates. That gives some information about that. And since this just came out today, I haven't had a chance to really interrogate it that much. Okay, so we'll go from talking about looking at cells, individual cells, new methods for looking at them, to methods for adjusting their functioning and analyzing that. So I don't know how many people know about this. Optogenetics. When I started my PhD, I went to the Society for Neuroscience conference in I think 2009, and this was like everybody was talking like, oh, this is the new thing. This is going to replace everything. This is great. And like the guy, Carl Deseroff, who was very much involved in inventing this, was kind of like a rock star at this conference. Like people were like, oh my God. And apparently it's only gotten worse. He's in this article I was just reading, he's like being accosted at airports and stuff by neuroscience people for inventing this. So why is this such a big deal? Right, because this is a big deal because using light, we are able to monitor or affect the activity of single cells in the brain. So this rat here, you can see this fiber optic cable is implanted into his brain. It's targeted towards a very specific population of cells. And by turning on the light, they can either stimulate those cells or silence those cells. So you can get some very precise things, which we'll talk about in a second. So all of this is based on these proteins called opsins that were isolated from bacteria and algae and things like that. And some of them respond to light. They respond to light by making some sort of signal, an electrical signal. And that signal can be used to either turn on or turn off the cell. So when these proteins are injected into a virus that's targeted towards specific cell types, and then that virus is injected into the animal, you can use lasers to either control neurons or monitor their activity, which is very cool. So by applying light, see, these are neurons spiking, you apply the light, or decrease activity, turn it back off. Depending on the kind of proteins or exactly the compounds and viruses and things you're using, you can also leave off. So that's cool to me as a neuroscientist, but what does that mean? Right? What does that mean? What kind of things can we do with that besides just make mice with light antennas coming out of their heads? Well, this one group has used them to what they call creating a false memory in the hippocampus. Right? So that's a big deal. We're creating false memory, kind of. So this is an example of things you might have heard about in kind of a more hyped setting than what actually happened. So what they did is they put a lab rat in a box, which is a typical thing. Neuroscience are doing that all the time. They're putting rats in boxes. And when they did that, they used optogenetics they identified which cells in the hippocampus were firing, a specific population of cells. So these cells are related to the place information. So those cells firing together or in a certain order correspond to this place, this box. Okay, so they have that. And then they train, they put the rat in another box and start shocking it, okay, which is another thing neuroscience people do often, is they shock rats, right? And while they were doing that, they were stimulating those cells that are associated with the other box. Okay, so they're associating shocking, this aversive kind of negative stimulus with this other place. right? So then they put the rat back in the box, didn't shock it, but it exhibited the behavior as if it was afraid of being shocked. So they kind of implanted this idea that this place is associated with bad things happening. So it's a very tiny kind of association. It's not quite, creating a false memory in the way we tend to think about memory, like this rich kind of experience of activities and surroundings and sounds and smells and things. But they were able to create an association where none existed previously. And people have done the opposite. This is very kind of technical, but by essentially doing the opposite of this, by silencing some cells, they were able to get the rat to forget that a place was associated with shocks. So it's memory creation in the tiniest kind of micro sense, and then memory deletion again in the tiniest kind of micro sense. And these are rats, not humans. And the kind of new theoretical frontier of this technology is, in addition to being able to stimulate and silence cells, you can also monitor their activity, right? You attach the protein to a cell, and then you, instead of shining a laser on it, you put a kind of photodetector there, and you can see when the cell is doing a particular thing. So the kind of new frontier, the technology for this doesn't really exist yet, but people are thinking about it, is using a kind of a closed loop, which means you are not only monitoring the activity, but based on the activity that you are monitoring, you are affecting cells. So you can kind of alter neural dynamics in real time is the idea. So we can put rats in boxes and get them to remember and forget based on a closed loop. Like I said, it doesn't really exist yet, but it's very interesting to think about this this system. But it's also very memory manipulation on a very microscopic kind of scale. We're again talking about rats and associating two stimuli together. So neuroscientists who work like this consider that memory. You might not consider that memory. I might not consider that memory in the same way, but it's pretty cool and a little frightening. So we're getting to the wearable devices. In terms of stimulating the brain with light, we're not there yet with humans, but there is certainly a lot of research with applying electrical activity to the scalp. Our brain is, an electrochemical computer. I guess you can use that analogy. But so people since the 60s have been investigating how we can apply minor electrical current and to our scalp non-invasively to affect our behavior. And like I mentioned at the outset, the actual evidence for this working is very mixed. There's some evidence that it could be helpful possibly in relieving things like depression, but only under very specific circumstances, under the care of a medical professional. Right? But that has not stopped this industry of people developing technology to sell to people. So this headset here, it's a transcranial direct current stimulation focus headset marketed towards video game players. You put this on your head, apparently it shocks certain areas, and you get better at playing video games. That's literally what they're marketing this for, and it costs a couple $100s. I don't recommend that as a purchase. I don't think you'll get your money back on that one. The issue with this is not only can applying electricity to your scalp be really dangerous, but just putting a thing on your head, if you're trying to stimulate electrical activity in very precise locations, if you don't have like high resolution brain images of your own brain and you're not very careful and there's not a doctor there, you don't know what you're really doing. So you're kind of like behaving like Ghostbusters. Like you might as well just put a thing on your head and start electrocuting yourself. And there are actual reports of people burning their heads, getting migraines, going into having seizures, all kinds of things. And there was a research report that came out saying that it's like instead of making you smarter or less depressed, this will actually make you less smart. I don't think that's actually true, but yeah.
Is there any regulation of these?
So no. The short answer is no. People have petitioned.
Can you repeat the questions?
Sure. So the question was, is there any regulation of these devices? And the short answer is no, not currently. People have petitioned the FDA to start regulating this and start saying you can't market these medical devices as medical devices because there's no actual research on this and it's dangerous. So pretty soon, hopefully, they'll be regulated. Right now, they're about as regulated as like nutritional supplements, that kind of stuff, where it says like not FDA approved, but then in bigger letters it says we'll fix everything in your whole life. So, at best this will do nothing. At worst, this will actively harm you. So I don't recommend any of these devices. They look silly and they don't work. If you want to, apply electricity to yourself, you might as well just go buy a 9-volt battery and put it on your tongue. Like it does basically the same thing. You have a question?
I've seen you talk about this TDCS, and the talk that I saw of South by Southwest, they were sort of raving about this DIY cell therapy.
Yeah.
And essentially they were saying, go get an 9-volt battery and go to Radio Shack and buy stuff. I guess it could be used in research on the brain or whatever. I guess it's just a comment. It's funny to hear the others that they sort of claim to that there's no reporting injuries.
Yeah.
Obviously promoting.
Right. So the question or the comment was that there are presentations at places like South by Southwest, and I'm sure there are presentations around like venture capital people in Brooklyn talking about this as being very effective, and this whole DIY brain stimulation movement has kind of sprung up. And that's very much the case. It's not uncommon for me to get like harassed by these companies on like Twitter, like, come look at our products and evaluate how awesome it is that we can affect cognition and emotion and all this stuff. And it's all advertising, basically. If this worked, like doctors and all kinds of people would be very excited about this. I would be super excited about this if it worked. I wouldn't have to spend hundreds of dollars per hour per subject to do brain research, like neuroimaging research. I could just buy a $200 headset and use it on everyone, right? They don't work. There's also an industry I'll talk about closer to the end of the presentation about like these apps for your phone or your Google Glass was big on this of like detecting emotion. Like you take a picture of somebody and you can tell what emotion they're feeling. Those things don't really work either. They're all based on like very overgeneralized principles of how all this is working. If we could shape memory, that means we would have to understand how memory works in the brain, which we don't fully. So, be a critical consumer of these things. Was there another question? No. Okay. That being said, there is some cool research being done in recording activity in brains and making it kind of quantifiable to the point where this hasn't really been replicated, but it has been reported in academic journals where this group had somebody look at just a visual stimulus, and they recorded their brain activity with an EEG cap, basically emailed it to a group who were using a brain stimulation machine called RTMS, transcranial magnetic stimulation, which is kind of the same thing as applying electrical current, but it's using magnetism, so it's a little bit safer. You won't burn yourself. And by recording activity in one person and by very precisely stimulating another person, they could have this guy who's blindfolded report when this person was seeing a visual stimulus better than chance. So it's kind of this brain-to-brain communication, very early, very primitive. When this came out last year, people were like, we can do brain-to-brain communication. We can have two brains communicating with each other. Well, I'm doing that right now. Right? My brain is communicating with all your brain. I'm just not emailing it to you. So this is kind of a cool frontier of where this could go, but it's very far away from being a thing that we can't. We're very far away from being able to download something very rich into your brain. So a visual experience, you have to wear a VR headset or something instead of it being beamed right into your brain. Is there a question?
Yeah, we're talking about magnetic stimulation.
So it's the question was about the magnetic stimulation. So RTMS is a way that neuroscientists have been using to disrupt brain activity in human subjects. They've been doing this for a decade maybe. And what's nice about RTMS is it's very time controlled, so you can turn things on and off very quickly. The most typical thing, I won't show the video because I'm not connected to the internet, but you can watch videos on YouTube of people being stimulated with this machine while they're speaking, and in the middle of a sentence, they will start saying a bunch of word salad, right? They'll be saying something very cohesive, and then they're stimulated, and then it will just be nonsensical. And they're stimulating speech centers, right? And this is a way of neuroscientists investigating circuits by disrupting them, seeing what happens when you poke things, basically. And the nice thing about RTMS is because it's using magnetism to disrupt the functioning, it's not invasive. You don't have to open up the brain. And as far as we know, it's not, it doesn't cause brain damage. You're doing this at such a time scale that you're not actively hurting someone's brain. It's about as the same kind of physics involved as an MRI, basically, just kind of in reverse. And people are starting to use this for kind of therapeutic things instead of putting electrodes on people's heads or doing ECT, electroconvulsive therapy. They're using this magnetic. Yes.
There were two other experiments that I remember hearing about.
Mm-hmm.
One was with either it was rats.
Yep.
And the other was somebody moving their arm.
Yeah. So this same group did the arm study. So the question was about these other experiments, and I think there's actually been 3 experiments. So there's been one rat communicating very basic information to another rat. There's been one person moving kind of another person's arm. And then there's been a person communicating with a rat, apparently. I don't know how that one works so well. I think they're question? Okay. I don't know how that one works exactly, but it's all using the same kind of recording stimulation kind of technology. What was the question?
There's also another one, not as far as communication goes, but as far as tapping into what the brain's subconsciously processes, where the experiment with the chimpanzees where you can play with numbers and you can tap the order of numbers. Mm-hmm. There's an experiment where when they interrupt the brain impulses, humans are more likely to get the pattern right when it's flashed.
Yeah. So
consciously aware.
Right. So the question was about these experiments into kind of consciousness and subconscious processes happening using this technology. And this technology is actually used a lot in consciousness research, people investigating our conscious awareness of things. So is our brain aware of things before we are consciously aware of it, that kind of thing? As a cognitive neuroscientist, those questions give me big headaches. And I think our brain is us. How do we know things before we know things? You can get very philosophical very quickly with some of that.
That's easy, but this works by use of it doesn't work by
It is magnetic. I don't remember exactly the physics of this because I did MRI, not TMS research, but it's using a magnetic stimulation to affect the electrical activity. So yeah, it's not applying direct current, but it's affecting the electrical activity for sure. Yeah.
The difference between powering a device directly into a wall versus sending out an EMP, which disrupts the entire.
Right. And you can also use this to stimulate as well, depending on frequency.
Magnetic resonance is affecting different levels. It's not
Right. Yeah, I could spend a whole hour talking about how MRI works. So if we scale up a bit from looking at individual cells or even kind of these discrete processes, there's also a level of neuroscience, and this is the level of neuroscience that I work in, where you're looking at brain regions interacting with each other, or you're looking at kind of bigger scale anatomy. So this is an image of the white matter tracks of the human brain. You can see that's very complicated. There's lots of connections going throughout the brain. And then there's a big movement right now to create the human connectome, which is adding "ome" to the end of anything is kind of like the buzzy science thing to do right now. You'll hear about the human connectome, bacteriomes, transcriptomes, biomes, microbiomes. And the connectome is basically an attempt to make a wiring diagram of the brain, how all the cells connect to one another. And that is a very, very difficult task because, as I said before, there are billions of cells and there are trillions of connections in the brain. And my set of cells and connections are not exactly the same as yours. We obviously share some similarities, but it's very different. And also, when you scale that up and just look at the interactions of kind of larger scale anatomical things, you also get very interesting connectivity kind of data. So this is C. elegans. This is a worm that is used a lot in neuroscience. Over at Rockefeller, there are a lot of people who work with C. elegans. We actually bring in high school students sometimes, and they do experiments with C. elegans.
Can you step back and forth with it so we can see the whole slide? Yeah.
Okay. Yeah. Sorry. And what interests neuroscientists about this organism is the fact that it only has 300-something neurons. So I'm not going to say it's easy to make a connectome of C. elegans, but it's multiple times. It's very much easier to do that than the human brain. 302 neurons is a lot easier to map than billions, right? So this is the Open Worm project. It's an online kind of collaboration between a whole bunch of different labs where they have created these computational models of C. elegans and all of its connected neurons. And this is amazing sounding, and it's led to some really cool things. But the work that went in behind this happened in the 80s. So we've had this complete C. elegans connectome for quite a while. But now we can put it in a computer and make very intricate maps of it. And this hasn't been reported in a scientific paper yet, but a group involved in the Open Worm project made I think it's the Connectome engine, I think that's what it's called. They basically put all this data into this little robot here and they just had it respond to sensory information. And it responded kind of like C. elegans, the worm, but it's a robot. It's all digital. So they made some noise and it kind of reacted. Obviously, it has equipment that's feeding it information about the noise level and everything. But they put it in, it moved around a room. It's actually kind of freaky to watch. You can watch videos of this on YouTube.
Did it learn?
I don't think so. They were doing very basic things with it. They were like ringing a bell and it was responding to that. They were putting down like chemical things, having it approach like it would in kind of the wild. I don't think they've done very much in the way of like learning experiments. What they did do is apparently they did virtual surgery on it. So, you know, it's a big tradition in neuroscience to do awful things to brains, right? To cut pieces off and see what happens. In the 1800s, we were doing that with animals and people, and we don't do that with people anymore. We just look at people who have pre-existing brain damage. So what they did is they like cut out some of the neurons, they deleted some of that, and it behaved like a worm would if you did the same thing. So they've done that. Again, this is all kind of one group who's doing this, not necessarily published anywhere yet. You can watch videos of a robot C. elegans on YouTube, which is pretty neat.
What worms was sensitive to smell but not others?
So they, I don't know too much about C. elegans. They work with humans, right? But they did, somewhere in this robot is a chemical sensor, and they laid down chemical traces that would be kind of analogous. And I guess it must respond to sound because it might have a very rudimentary auditory system. I'm not sure. So to go from a C. elegans to a rat is a gigantic jump, evolutionary-wise, but also in terms of connectome, right? So this is a very simplified version of different parts of a rat's thalamus and cortex, with all the connections between different pieces. So you can see, I mean, this is a very complicated looking dots. You can't see the lines, but they're all connected to each other. This is just one brain region interacting with the cortex. I mean, it gets substantially more complicated is the message here. This isn't even cells, this is regions. These are groups of cells. And in humans, this is like a terrifyingly complex thing to try to create a wiring diagram of the whole human brain. Frankly, the reason we aren't doing it is because there is no computer powerful enough to do all of the calculations necessary. I'll talk a bit about a group that's trying, but this organization, iWire, they started out as part of MIT. I think they're an independent group now. They're mapping the retina and the connections of cells in the retina by leveraging the free time of people playing video games, basically. So it's a video game that you can log on to and play, where you're basically mapping connections between cells. So they're leveraging like citizen science, basically, to get this to work, because we can't develop a computer to do this. So we'll just use video game players. So if you want to contribute to neuroscience and you don't want to like get into a lab and, you know, squish some brains or whatever, you can just log onto your computer. It's free. It's kind of fun.
I saw someone give a lecture at a citizen science conference.
Yeah.
And they've actually done a lot of work to make it attractive.
Yeah.
So they have all sorts of, you know, sort of rewards and levels, and they made it. They really tried to make it like a very compelling video game. And they redesigned the website several times so that it was very easy to drive traffic into it and register.
Yeah, when it first started, it was kind of clunky and not very fun.
Yeah.
Now there's like people who are top score iWirers, and they're, you know, it's like every kind of video game. There are people competing with each other, and there are different groups, and people are getting really into it. So it's this leveraging of this, you know, cognitive space that people have to solve this very complicated problem.
Do you yield any scientific outcome that's verifiable?
So the question was, has it yielded scientific output? I have not kept track of their publications, but it has generated, like they've created maps of connections. I do know that, and that they do publish. So I'm sure it's not just kind of a vanity project for neuroscientists. I think it's actually yielding a lot of very good data. Question?
How are they gathering the samples from these tests?
So it's, I wrote it down. It's they use electron microscopy on actual tissue, and they made three-dimensional images. The problem with electron microscopy is it's black and white. So basically people are using, yeah, exactly. So how this works is they have slices from the electron microscopy, and you work within a slice that's randomly assigned to you, so there's no bias, and there's all kinds of ways of connecting and working. It's pretty cool. I think this was featured in Times Square recently, actually, like on one of the billboards. So the Human Brain Project, I mentioned it when we get to it, and we're getting to it. So Human Brain Project was conceived as the European version of the Brain Initiative. And what ended up happening was it got kind of cornered by this group who wanted to make a computer simulation of the brain. And that's a very exciting prospect. I heard the director of this group talking recently at Rockefeller, maybe a couple months ago or maybe a year or so ago. He was talking about how they were going to continuously make better models as better supercomputers came out, and they were working with all these computational people to make this work. The problem is the data for making a simulation like that doesn't really exist for the whole brain. And also, all of the money was going to a very small amount of groups. So basically what happened was European Neuroscience kind of rebelled against this and said, we can't have all of our funds going to this one project. That's completely ridiculous. They were cutting out funding for research that I was doing, these larger scale projects. So right now this is kind of in a set of stasis. So many people signed this open letter saying that this was not well conceived, this has not been well implemented, this is just for a tiny group of labs, but this is kind of at a standstill right now. So there's not a lot of forward momentum in terms of building a human simulation of the brain. Mostly because that's a and this is a circular connectogram of the human connectome. So I think that's as many buzzy words as I can possibly fit into a sentence. But what this shows, it's impossible basically to read all the figures here. This is a map of the interconnections of the human cortex. So as you can see, it's massively interconnected and massively complex. This came out a couple years ago, 3 years ago, and we know that it's actually even more complex than this is showing. So
how do you read those kind of graphs? Different inner rings and outer rings?
So I cut out the legend, so it's impossible to read right now. But the labels are all labels of individual brain regions, like anatomical regions. And the different colors correspond to different groupings, different networks, and things like that. It's different functions, possibly. I cut out the legend, so basically, it's just to show everything is massively interconnected. There's things, regions are connected within. There's intra and inter connections. Very, very difficult. And this idea of the human connectome has really kind of permeated neuroscience to the point where it used to be, even 10 years ago, 15 years ago, when people talked about functions in the brain, they would often default to talking about individual regions. So this region does this. Hippocampus is memory. Dorsolateral prefrontal cortex does X. Medial lateral does Y. Different designations for different areas. And when I was a student of neuroscience, when I was an undergraduate and doing my doctoral work, like it was emphasized to us over and over and over again, like that is not how the brain is anatomically, so stop doing that. And that finally seems to be kind of hitting home now when people talk about disruptions in the brain. They're talking about disruptions in kind of networks, so connected regions. So instead of looking at just this region here, they look at this network here and disruptions between connections. So we've gone from kind of a modular view to a network view, which is good because that's how the brain is more or less wired that way. There's nowhere in the brain has one function, and no one function can be localized to one region. And this is me just talking about something vaguely related to the research that I did in grad school, but the simple act of looking at and processing a face. So if you were to see a picture of a face, or a face kind of in the wild, if you're looking at somebody's face, it activates a very wide distributed network of brain regions. Individual areas, so back here, the FFA is called the fusiform face area. It's a fusiform gyrus. And in addition, it also leads to activations and activity in places like the amygdala and the prefrontal cortex that are associated with kind of social things that they tend to be discussed in terms of social functions. So this is a very simple thing, right? Looking at a visual object activates this whole kind of distributed network. For my dissertation research, I literally put people on an MRI and showed them pictures of faces and looked at these regions. And that's a very simple thing that they were doing. They were just literally just looking at a face and saw this whole thing. And this has kind of been growing in popularity since I was in graduate school. It's kind of exploded now that we have the Brain Initiative. This idea of a default mode network. So this is brain regions that are more active when you're not doing anything. So this is your default mode. Kind of the idea is you're just resting. So you're at a resting state. Often when people do brain imaging research, they put people on scanners and they have them do a task. I had people look at faces, and I had people look at scenes, I had people do simple math tasks. People are usually just looking at a crosshairs on the screen. And by looking at resting connectivity data, so you can look at brain activity while they're ostensibly doing nothing, you can map connections by these activities. And then if you have two groups of people that are different, you can look at differences in this default mode. And this has been investigated a lot in terms of mental illnesses and cognitive capacities. There's not a lot of firm research showing that one group has one pattern of default mode activity leads to one outcome versus another, but this is very much kind of happening, this type of research. And mapping these networks is a big part of human connectome research. Yes?
What's the biggest difference between men and women in terms of connectomes?
So this is my kind of sarcastic but not sarcastic response to any question about between group differences. There's way more difference in between people within a group than between groups. So, to There is, of course, a lot of research looking at gender differences in neuroscience. But I ran experiments with male and female participants and put them all in the same group. And you can't look at a brain active or resting and say this is a male brain, this is a female brain. Yes.
What about face blindness? Can you tell if a person is face blind or not?
Yes. So there is. Can you tell if somebody is face blind by looking at their activity? So with this face network, if somebody has prosopagnosia, which is face blindness, they show disrupted activity in those regions, and oftentimes damage in those regions is why they are face blind. So that kind of thing you can detect. There's some research looking at autism and things that are have symptoms that are social based, looking at these regions, but there's nothing definitive really. But getting back to the gender thing, there's obviously some potential for hormone differences in male brains versus female brains, right? But besides those, there's no difference that you can detect with this stuff.
What about the bigger corpus callosum and women? I believe that they have a bigger white matter connection between the two hemispheres.
So the question was about the corpus callosum, the white matter tract that connects the left and right cerebral hemispheres. And I can't speak to that research because I haven't looked at it in depth, but it's again one of those things where if the average is slightly different between these two groups, it's not that you can look at a brain and say, oh, they have a larger corpus callosum, that must be a woman, right? Because that's an average of those two groups, and the intra-group variability is such that you could look at my brain, you could look at my wife's brain, I could have not told you which one was which, and you would not be able to tell. Maybe you could tell by like the head shape and the fact that I have a gigantic head while my wife has a normal-sized head, but you can't really look at it. Question?
Yeah, I think that the problem is basically experimental design. If you want to show differences between men and women and left and right, you don't show them faces, you show them Jimmy Choo shoes.
But that's really the difference between culture, because at the same time, you can have, say, I don't know, show somebody from Thailand a bucket of like pork, sure, like delicious street food. That's the same thing in Thailand. No joke. So then they're going to that be like, oh, hey, that's going it's the same thing. It's
Yeah.
cultural. It's cultural about the second.
I was actually just kidding.
So the question was about looking at these differences using tasks in the scanner, and my problem with all that research about gender differences or looking at the brains of gay versus bisexual versus straight people is so what, right? Like if you look at a male brain versus a female brain and you see a difference in their activity when they look at something, what does that mean? It means you found a difference, but it doesn't mean like one is not an abnormal type of activity versus another. It's just different people who are different and have different brain activity. So ultimately, just because you can look at some differences doesn't mean they're actually explaining anything. Question?
Yeah. How do you generate what
So that's a good question. Whose connectome are we mapping, since there are so much variability between people? There's two ways that the Human Connectome Project is solving this. First way is scan a ton of people, look at the connectomes of hundreds of people, and average them together. You get kind of a model that is an average. Another group, I think they're Harvard and Boston area people, are making very high resolution scanners, so you can get very high fidelity images of individual connectomes. So it's like average of multiple people versus a really good scan of one person. And these two groups are interacting with each other, so two approaches that are hopefully going to converge, but we'll see. The problem with this type of research in general is you have to do a lot of averaging to get this kind of image. My dissertation work was looking at faces in people with depression versus non-depression, so healthy versus depressed, and people would always ask me every single time, no matter who they were, they would always ask, looked at my brain scan, am I depressed, am I not depressed? I can't tell that by looking at an individual brain scan. I have to average a bunch together to look at the possibility that these groups differ. So, and in point of fact, there's probably, again, more variability within each group than between. So, there was a movement for a long time to find endophenotypes. So ways of looking at a person's brain to say, oh, this kind of pattern of connections means this person is depressed. We've kind of moved away from that because we didn't find anything that was reliable.
In more time sensitive, like you connectors say what are
So the question was about kind of functional connectome. You're obviously feeling differently and thinking different things at different times throughout the day. Does that affect these connections? And what you're talking about is structural versus functional differences. Probably our white matter tracks stay the same throughout the day, but the regions of our brain interacting with particular patterns depends completely on what we're doing. And so one of the criticisms of this whole idea of a connectome is, well, okay, you made a wiring diagram of this network. You're not looking at the activity of that network and how it's operating as you're experiencing things, taking in information, putting out activity. So yes, very much the kind of functional interaction between the regions changes every second, right? Different conditions will affect these connections in different ways, but we're at a point where we're just trying to kind of put the network together before we start looking at the function. So that brings the whole principle behind these brain training games like Luminosity. There's a billion different versions of this. I could probably find a whole bunch in the App Store, right, if I look. Is that they affect connectivity and they help train your brain. They help different parts of your brain be more or less active. This is a scan that this is a brain image that Luminosity puts in a bunch of its advertising material. Like, look, we did something that made brain activity go up and down, which everything can do that, right? Like if you apply a low statistical threshold to your measurements, you can literally get brain activity doing anything. They didn't give any necessary data to interrogate what this means. So basically this is, if you do a thing, your brain does a thing, which sure, you could do that for anything. If I were talking to you, I'm sure different parts of my brain are lighting up. That doesn't mean I have been trained. It's craziness. So these games purport to affect your brain. And by having you do tasks that are basically slightly prettier versions of tasks we use to judge if somebody has brain damage, they say that they're affecting your cognitive ability and making you smarter and making you be able to recognize faces and do all these really amazing sounding things, and you're making billions of dollars doing this. The problem is there is no evidence whatsoever that these effects on the brain, if they're positive, that they last. And there's no evidence to support the widely held belief that the regular use of computerized brain trainers improves general cognitive functioning. Basically, by playing these games, you get better at playing these games. You don't get better at memory in general. You get better at this particular memory game, which is good if you want to be a competitive memory game player, I guess. But it's all based on a set of assumptions that aren't true or haven't been found to be statistically validated. Questions?
So my idea is, what does it mean for somebody to be smart? And one person can solve one math problem, the other one.
So that is the big question in psychology, and neuroscience, philosophy. There's no good answer to that, really. Like whenever you use a test to get a score for someone's intelligence, you are basically testing their ability to do that test. Right? And you can make a broad test or a narrow test to try to get a broader picture of what that means, but neuroscience doesn't really have a good answer to that question. It doesn't mean being able to remember more things isn't necessarily make you more intelligent, because one of the things that helps us get through the day is being able to filter relevant versus irrelevant information, right? So if you're just retaining everything, you might have a harder time. So unfortunately, there's not a good answer to that. And that's part of the problem with this, is we don't have a good answer to what is intelligence, so we can't say, that's not intelligence, this is intelligence. But what I can tell you is, these games will make you really good at these games. That's about it. Good question behind you? Yes. Right. So the question was, is there a way to prove that these games work? Basically, or don't work. Is there a way to falsify this hypothesis that these games work? And yes, and the way to investigate that is to train somebody on one of these games that is purported to look at memory, and then have them do another memory task before and after playing the game. You can do that with one person. You can have twins that are the same genetically and train one and not the other. However you want to do that. And people have done that, and that's how we know that they're very specific in what the effects are. You can also, let me start that again. The one thing I will say about luminosity is it's a really nice way to collect a lot of data about these tasks. And they've been very generous about sharing that data with people. So you're not training anyone to be smarter or whatever, but you are assessing their ability to do basic kind of cognitive tasks. And if they also give you information about their age and their various other characteristics, you suddenly have this gigantic data set of kind of psychological measures, which is interesting. People haven't really taken advantage of that because people are kind of weary of this whole enterprise, but they do collect a lot of that data that could potentially be interesting. The fact that they're not collecting that data under the auspice of their task doesn't work, it's kind of besides the point for that. Yes.
One is, could tests and games like that be useful for telling the difference in your mental state? Like usually, if you have a test like that, I think, like, even though they're not different for your body, could they give you good feedback on yourself?
Yeah. Okay, what was the other question?
Okay, the other question was like, what's your opinion about the neuroscientists game? Like, do they do it for the money, or like, are they just bad neuroscientists? Or do they really believe in it?
So the first question was, can you use these things to kind of assess yourself or assess one individual? And the answer to that is very much yes. These games are all based on tasks that were designed to do that exact thing. So a lot of these games are based on things that you use to assess people with brain damage, or some of these tasks are used in studies where they have someone do the task and then they have them drink a whole bunch of alcohol and they try to do the task again, and you can measure how their performance has decreased. So you can do that. That's sort of what they're designed for, actually. And in terms of what do I think about the neuroscientists behind this? So luminosity, I know, was started by somebody, I think he left graduate school for neuroscience or something like that. There are neuroscientists kind of working behind the scenes of these things. And I'm going to be charitable and say I think that they think they're improving things, right? I think, They probably left academia for private enterprise because there's more money in that. But I don't think, I hope that they're not sinisterly like, we know this doesn't work, but who cares? We're going to pretend it's neuroscience. I'm being charitable, I think, but I would think that they think they're doing good stuff. And a lot of this was investigated, in the last few years. People are very excited about the prospect of brain training people to work through PTSD or different things, but it hasn't really panned out. So the joke about academia is new ideas won't go away when all the people who believe them die. So people probably are holding on to this idea, one, because they have a financial interest, and two, because they just think that they're yeah. Any other questions? Yes.
There's, I guess for some time, there's been this connection between space and ability.
Mm-hmm.
You see this anecdotally,
Yep.
stories, whatever, and I'm wondering whether with your imaging data, you see that relationship between spatial perception and
Yes. The short answer, okay, so the question was, what is the relationship, or have I seen a relationship between spatial recognition or spatiality and memory? And if you look at the neural architecture, the parts of the brain that are widely kind of investigated for their role in spatial navigation are tied very closely into those that have to do with memory. There's kind of these anecdotal, not so anecdotal studies of looking at the hippocampus of regular people versus London cab drivers who have this intricate mental map, and looking at differences between those two things. And the short answer is yes. In my own research, actually, we showed some people scenes of outdoor kind of scenes, so like grasslands and different things. And we found that depressed versus non-depressed people had some differences in the activity level in those regions. We were like, we just couldn't figure that out. Like, why would someone who's depressed perceive a scene of grass differently than a non-depressed person when you would think they would perceive faces differently, and they don't seem to be doing that. And what we figured out is probably there's just some memory difference in general that we're picking up in those regions. So the hippocampus is widely a memory area. The parahippocampus is often the spatial navigation area. And so there's a very intrinsic connection between those things. We probably evolved our brains evolved while we were running around through forests and things. So it's all built on this architecture of spatial navigation. Yeah.
Is there any way to explain phenomena like my boyfriend and I will go see the movie. I'll remember the plot of the movie a year later. He won't remember it a day later, but a year later he'll remember exactly where we parked in the parking lot.
Yeah, so the question was about remembering kind of different things, like the plot of a movie versus where you parked to see that movie. And I used to think about this a lot when I was in graduate school. I'm like, I can't remember all this neuroscience stuff, but I could tell you all this ridiculous Star Trek trivia. It's all like in my brain. I don't need this. I need this other stuff. There's not an easy answer to that. Why certain people are better at remembering certain types of information versus other people. It probably depends on the salience of that information while it was being encoded. For whatever reason, maybe your boyfriend his brain is wired to remember where the car is versus the plot of the movie, while yours is wired more to the beats of the story that you're absorbing. And that's just kind of conjecture, right? I'm just making that up. But there's no easy answer to that.
Do you think depression harms spatial memory?
The question was, do I think depression harms spatial memory? So I'm going to give two caveats, or one at least. Whenever I talk about depression or any of these things, people start asking me questions like, does depression do this? Does depression do that? Depression is a hugely heterogeneous condition that affects different people in vastly different ways. problem is our definition is symptom-based, not neuroscience-based. So looking at the neuroscience gets very tricky. And the other caveat is I'm a cognitive neuroscientist, not a clinical psychiatrist or psychologist. So doctors versus doctors. I think as somebody who studied neuroscience as an information kind of processing condition, and studied depression, depression affects information processing globally. We tend to think about it affecting our mood because that's the symptoms we see, but if we put somebody in an MRI and look at their brain while they're looking at very innocuous stimulation, you see differences. Their performance is the same, so there's different kind of compensatory kind of things happening probably. I think it affects kind of information globally, maybe more so if it's got some emotional component to it, but I think it's global. I think that some of the data that's coming out is starting to show that a paper published right when I was leaving graduate school showed that the perception of contrast gratings, so like this very basic visual pattern of lines basically in this direction versus this direction, people with depression versus healthy people were showing different brain activity in response to the same stimuli. And that's completely innocuous, very basic stimulation. But that's just information. And they were seeing this at the level of like, retinal activity. So if the question is, does depression affect X type of information, my answer is usually yes. How that manifests in terms of symptoms, I can't speak to that.
When you go back to the games.
The games, yes.
And how to do that. Probably I remember listening to on the radio to the Neuroscience team, and they were saying, download the Neuroscience app about that.
Yeah.
By the end of the commercial, I have already forgotten the name. I have to listen to it like 5 times. I don't know that now. So I thought I definitely need it. So this is probably wrong because they were saying, it's going to improve my memory in general.
Yeah.
But, I think if you prepare playing these games to watch TV, or if, for example, I read recently dancing, it's great even to prevent neurodegenerative disease, and the whole thing about brain exercise. What do we know? How do we get
So the baseline to these studies usually is someone doing this task versus someone Googling stuff on the internet. And there's no difference in terms of what the outcome of those things. But one thing I can say that doing these things won't hurt you, and paying attention to your kind of cognitive state and your memory and brain kind of health is a positive thing. And if you're doing something that's exercising your body, that's going to be beneficial to your brain. So we know that. We don't know exactly the mechanism by how that works necessarily. There's lots of theories about it. But if this has a positive effect on a person, it's because probably they're paying attention to their attention, they're paying attention to their memory, they're kind of thinking about thinking, which is a good way of improving your thinking. Or it's a placebo effect, and if it works, great. It's not harming anybody to play these silly little games on their phone on the train. So, I like to poke fun at these games because the evidence isn't really backed up. If it's working for somebody, I'm not going to deny them that. So the question was about the London cab driver study looking at brain of London cab drivers versus non-London cab drivers, whatever the control group was. That study, there's like this whole kind of cottage industry in neuroscience about looking at London cab drivers. Like if I was a London cab driver, I would be scanned 1 million times probably, more than I was scanned in graduate school, which was a lot, let me And the effects are reported as being significant, but they're kind of anecdotal, and these sample sizes are very small and very specific, and there's often just sampling issues. So it's a fun thing to say, and it's a good example to give of like, look, this structure can be affected by function. These things can affect things that we can see, but it's probably much more complicated than that. It's too early to dismiss it, but it's also too early to kind of accept it wholeheartedly. And these games are a lot less. The detail and the type of thing that you're remembering is a lot less rich in these games versus a street map of London. Like, that's an intensely complex mental map that you're building. These are like connecting dots together, associating different cards with different things. Yes.
There was a time when these scientists were doing this famous book of Persuasion of Man by Stephen Wearable. All these things, is wrong. Is it again coming back up, or this academics have got promoted and no more such management distinguish between black people, white people, red people, women, men, etc.
So, unfortunately, yes. So the question was about looking at different groups, different social groups, or different groups of people, different races, different ethnicities, different people with different gender identity or sexual identity, and looking at trying to parse that apart biologically. So neuroscience has a very troublesome history with doing that stuff, as I think we can all imagine, and science in general, but neuroscience in particular. The ethics in neuroscience came after some really horrible experiments. So I would say the majority of neuroscientists are very averse to doing that. People who look at gender differences even you'll read a lot of caveats about that if they're being responsible, which most are. But there are people who are, unfortunately kind of hedging very close to looking at genetic differences between. The example that springs to mind is a group that I know of that's looking at genetics and socioeconomic status. And that research hedges very close to these people are poor because they have this set of genes, which is a horrifying idea, and one that's obviously false, right? And so the nice thing about modern neuroscience is that it has grown up alongside neuroethics, which is emerging, and people are cracking down on that. Even if you find a difference, it doesn't mean it's meaningful. It doesn't mean one group is better than the other. It doesn't mean your genetics aren't your fate, right? You can overcome whatever biology has thrown at you.
Sure, it could be something as simple as people from the same socioeconomic class who marry each other.
Right. Yeah. I think one of James Watson, I think,
Yes, Watson was
was in the news recently because 2 years ago by saying some really horrible things about genetics and social classes and social groups. And it came up again recently when he was trying to sell his Nobel Prize because the scientific community has basically shunned him because of these horrible, terrible views. So there are people working kind of around that space, and I'm sure there are people who are using that work to support their horrible ideas. Just people latch onto anything. Yes, it's a very small minority.
Using human, whatever you want to call it, science or whatever, to that evidence and or get you to understand aspects of is this true? Really happening?
So the questions about neuroeconomics and neuroadvertising, is it happening and is it true? Those are different questions. The first question is, it is definitely happening. I think this year there have been several hundred patents for this kind of technology to look at neuromarketing is the term given to it. Nielsen, like the big market research company, owns a lot of these patents. Does it work? And does it, is it saying what they say it's saying? I don't actually think so. Often I have a friend who works kind of in a space that's tangential to that. And she was saying, often people will show data like, oh, look, the hippocampus or the amygdala was activated when they looked at Coke versus Pepsi, which is a ridiculous statement to make, because what does that even mean? Like, they say, oh, it means they're more excited about this versus this, but that's not what that means. That means any number of things. These regions have many functions. And I'm guessing that people doing this research are probably not expert neuroscientists and nuclear physicists and things. So there's a lot of money there. There's not maybe a lot of science there.
My comment about whether or not it works is one of these things that we don't really have to put science to. All the regulations that are communicating with the brains.
No, exactly. So the comment was, the comment gets to a point that I bring up a lot when I talk about this. Just because we can pretend to use neuroscience to explain a thing doesn't mean that we have to do that. Asking people, do you prefer this versus this, is as effective as looking at their brain. It's more effective than looking at their brains, and it's a lot cheaper.
It's just the same as we talk about people with better members from other teams. We'll just ask what their interests are and always
exactly.
their interests.
The reason it's so popular is because Nielsen or whoever can charge their clients lots of money. So, yes. Getting to this question of neuroeconomics, though, there is some interesting research going on. People are having people do economic decisions and looking at the dynamics of their brain, like how people evaluate risk and things like that. That's an actual area of study, but it's called neuroeconomics because it gets funded by economists, but it's just neuroscience, risk perception, and stuff like that. And economists fund things more than neuroscientists. Let's skip some stuff and talk about multivoxel pattern analysis in neural decoding. That's kind of the last thing I'll talk about. So this is a technology that is really cool and was coming into its own when I was finishing graduate school. So this is, you take a brain scan of somebody as they look at stimulus, look at a cluster of voxels, and so a voxel is a three-dimensional pixel, basically, in the scan, and you train some sort of algorithm to associate a pattern of voxels with individual stimulus. So this example here is, this pattern means apple, this means banana, this means apple, this means banana, and you feed all that into an algorithm, and you can then look at scans and classify kind of what the person is looking at, which is pretty neat. So there's an example of what this has been used for is, for a long time, since 2001 or so, there's been this whole social neuroscience idea that rejection, like social rejection and physical pain, are kind of the same thing. They activate the same part of the brain. Like, does rejection hurt? Is physical pain and social pain the same thing? When someone breaks up with you, is that like, are they physically harming you? And so somebody looked within this cluster and found that this is the anterior cingulate cortex, which is important because this region is activated in basically every study. So the fact that they found it in social rejection studies and physical pain studies doesn't really mean a whole lot, because it's activated in 50% of all fMRI studies in general. And they looked at these patterns for physical pain. They trained a computer to recognize these patterns, and they found that within these regions, different voxels were activated in different patterns. So what this region probably is like an attention. It's probably attention and salience. So this is an important thing that's happening. This is an information hub of some kind, not some weird like social rejection against physical pain kind of thing. There's a lot of attempts to make the brain to fit our kind of social conceptions when it's kind of a computer just analyzing information. a friend of mine sat in a lab meeting once where they were talking about the neuroscience of justice, I think, and I was just like, how? What? That's like a concept that we define, philosophically when you're looking at biology. It's a question. So whenever you see a scan like this, it's very often composite, or an average of I think it's just like 10 people. And these classifiers are usually trained in a small amount of people because to get this data it takes hours and hours and hours. So you can only subject like 5 people to it. It's usually graduate students in the lab, right? You have to do this. Your dissertation depends on it. Get in there. It's MRI.
So everybody's seeing the same box.
So the question was, is everybody seeing the same pattern for voxel? And the answer to that is usually these things are trained on individuals. Probably because the resolution is such that it's just easier to go individual by individual, and then you average their scores together. So you'll use a particular algorithm in a particular region, but the pattern will be different for people.
It definitely is. Because if you think about it, sorry, there's a couple of experiments. One which you might know, the kidney experiment, where these kids are engaged. They have a device attached to their hands, so they can't turn very good. They move from the lines.
Yep.
As they get to a certain age, they can't perceive that what's on the lines. So when it comes to these fossils, if they can't perceive them, there's literally no registration here. And being this person has neurons, small dogs, and we perceive this in the same way. That's why so exemplified human estimates. There's a village in Africa where they see their structures are circular, of course, whereas ours are all lined and horizontal and angular. These people who come to our country, they have not a difficult time, but they don't have as much precision in distinguishing the lines. The readings are much different as well.
So those are both good studies. That kitten study was done at Rockefeller, which I think about whenever I have my cats at home. No, stay here. Don't come to work with me. There are, depending on what level of kind of the visual parts of our brain you look at, though there is retinotopic mapping from what you see to kind of cells in your brain. That's pretty consistent from person to person. When you talk about perception, though, you start getting into philosophy of mind and start getting very far away from things that we can detect with fMRI. It's all a pattern, but it gets complicated. And this sounds really great. Like we can find these patterns in the brain. We can distinguish this versus this. Oftentimes we're just seeing if the pattern can distinguish one thing versus another thing better than chance. So the actual results of this are not as, we're using a lot of computational power to get something that's not as great as we would like to think it is.
What about, there's a video, I think it's a TED Talk, where a woman is showing a brain scan interpreted, and it's showing what the person is looking at, like an airplane, and you see a fuzzy image of something with wings, and I mean the resolution is amazing considering it's just a scan.
I have a couple of slides on that. I can tell you about that particular. So the question was, we'll get to that right now, actually. So you can use these algorithms to, once you know the pattern of particular objects, you can then look for that pattern and kind of reconstruct what a person is seeing. So how this works is you train a bunch of runs on looking at, in this case, different bees. And the algorithm learns like this set of voxels in this particular pattern corresponds to B. And then you look at a pattern in a brain, and the algorithm is naive to what's in there, and it reconstructs a B from that pattern. And how this generally works is they use an early visual area, so which is like V1, for example, our first primary visual cortex, where things are kind of projected onto the cortex in the way they appear on our retina, so you can very easily kind of reconstruct it. When you start talking about like faces and our perception and deeper kind of processing, that gets very tricky. So this group trained the algorithm to recognize different letters and then had it reconstructed the word brains based on the neural activity it saw in the person's brain. So decoding, it's decoding perception sort of from brain activity. And as you can see, it's not perfect, obviously, but it's pretty close. It gets tricky when you start looking at more complicated things. Not very far. So the question is, how long in the processing stream are we able to go currently? And the answer is not very. If you look at, this is faces. So a group recently tried to do this with faces. They trained the algorithm to look at a simplified version of faces and detect different visual features, and then reconstruct a face based on the visual features and the pattern of voxels. And as you can see, like, the results are not super great, and they kind of all blend together. And this is still in a pretty...
Do you say that's better resolution, precision, or accuracy?
So, a couple of problems. One, it's a matter of precision of fMRI, because our voxels are millimeters in...
That's big.
Yeah, so our voxels are quite big. And there's like hundreds or thousands of cells within each voxel. And using NVPA pattern analysis, you get a gross measure. And you know, we don't know where all these inputs are coming from. And by the time something gets further along in our visual pathway, it's not just, our representation of it is not just influenced by the actual visual information, interference around it, but also your kind of top-down process, your expectations of what's there. You know, you're kind of, you know, when you see the visual image, your visual experience is not matching the information coming into your retina, because there's a lot of interference. You have stuff in your eyes, you have blind spots, and you know, a huge part of our brain is dedicated to vision because it has to fill in all that information. And a lot of that is not guesses, it's kind of computation, but it's not matching reality. So in the sense of, you know, direct information coming in directly. So when we're trying to reconstruct a face, we're not just having visual information, we're probably getting information about non-sensory information, or expectations, things from higher areas, all manner of things. So this gets very difficult very quickly.
But it is seeing that you're looking at the face,
Yes.
which is absolutely stunning.
Yeah. So
It knows you're looking at a face and not an owl.
right. But under a very specific set of circumstances, when you've already trained it to look like this is what faces are versus this is what other things are. It's not that we could like pull somebody off the street and they classify and say, oh, you're.
So it's that individual specific.
Which has not stopped groups from trying to pick up on this technology. This group is called No Lie fMRI. That's the name of their organization. And they are trying to use this technology to look at lie detection. Can we tell, using this technology, if somebody is lying? And the answer is no, because what is truth? What is lie? What is perception? What is the part of the brain that's the truth area? What is a truth pattern? If someone believes something is true, but it's actually false, what does that mean? Like, you know, it gets very complicated. This is, again, another thing of like selling stuff that doesn't really say.
But what about a carefully controlled experiment? I mean, I know you mentioned that some of these things are very undefined. There's lack of understanding of what the space is. But in a controlled experiment where you have the definition of something that's always true, compared to something that's relatively false, fairly apart from reality, in that situation, yes, you're not actually comparing regions that are truthfully described and truthfully sure, but in that experimental setup,
So
it's a relative assessment.
Sort of. But does that, so what these kind of studies do, they do very basic truth versus lie type things, or truth versus deception, or incorrect versus correct information. So you can say to somebody, 2 + 2 = 4, 2 + 2 = 5, and look at the difference in brain activity. Right?
It's establishing baseline.
Right, which is, at that point, I'm not really sure what you're measuring. Are you measuring truth versus lie, or expectation versus, like, something expected happened, I know 2 + 2 = 4, so I expected that, versus 2 + 2 = 5, I didn't expect that, was wrong. So it gets very complicated. And outside of that, to use it for something as complicated as, like, did you murder someone on July 25th, it's completely useless, right? There's some.
In a simplistic experimental design, scores, spots, where clear distinction is what's right and what's wrong. How the ephemeral pattern lines up in that situation, when it comes to mind, what the brain interprets at this and might, for a very simplistic
Yes.
logic on them, like to.
Right. So the problem is, that's actually a quite interesting processing question. Like, how do we deal with information wrong versus right of that type? The problem is people extend it way beyond because they're trying to sell you stuff.
I mean, the question here is, what are the different behavior of the brain that is consciously saying true?
Yes.
Independently of that is true or wrong.
Right. So that the question was, like, looking at brains who are people who are consciously versus kind of unconsciously or unknowingly lying. And it's very hard to measure that. It's hard to put people in an fMRI and have them tell you things that are false, but they don't know are false. Like, just creating the experimental design for that is quite tricky. And again, that's not the same thing as lie detection. You're just looking at belief, information processing at a very basic level. So I have 2 slides at the end here. This is a piece of software. This guy, you can't really see, but he has a box around his face and it's registering surprise. This is an app that was for Google Glass. This is like some marketing for that app. It was supposed to detect people's like their identity, if you've seen them before and tag them, their gender and their emotion, and like give you, like, in case you're terrible at human interaction, I guess. Like this person is sad because they're crying. You know, and this is all based on decoding of a different type, not decoding brain activity, because you can't look at someone and decode their brain activity, but it's a similar principle of trying to decode micro expressions. And the reason this hasn't really caught on is because it doesn't work. This kind of facial recognition software, like recognizing my face versus somebody else's face, that software is really effective when you can do that. So that's a basic visual question, kind of basic visual
They
information.
have an age one too. That's
I mean, it's error bars, that's not so bad. But some of that stuff is good. But this expression, my very first job in the lab was doing emotion research. And that area of research is very complicated because this person is surprised, I guess, but he could also be like bored or amused or any number of different things, and the expression is the same. And our description of what emotions are context dependent and language dependent and subjective. And so, you know, this stuff gets very complicated. And so if someone tries to sell you an app to help you do basic human interaction, it's probably oversimplifying. And probably based on, you know, these ideas about expressions that are very defined for a thing that's not very well defined. Like they're assigning very concrete definitions to things that are more loose. And this last slide, I just saw this today, like right as I was about to leave my desk. And it's neuroscience fiction. This is a group trying to sell you an EEG that you can wear on your head, which is actually kind of neat, right? To have a portable EEG costs a couple $100. That's cool. I like that. Like you can do neuroscience experiments in your garage, and you can subject people to things. That's cool, I guess. But this group is trying to market it towards like monitor the attention of students in class using brain imaging, monitor their cognitive power and like increase this wave and monitor that in real time. And that's complete nonsense. This technology is all based on the same thing that's like, there's like a Star Wars toy where you put on a thing and
We have one upstairs.
you have one?
Yeah.
It's all based on the same stuff. They detected very basic kind of EEG signal that's like attention or effort. It's very general. It's not the same thing as learning. It's not the same thing as training someone's brain. Like, oh, your alpha wave is too high right now, so go do this other thing and come back. Like, to me, this is like neuromarketing and neuroscience fiction of the worst kind, because it's like trying to target a thing that people are really into. Like, education is a big thing, and trying to understand how to get people to learn is a big problem. And it seems like if you don't know that this is nonsense, to apply like this biology to this thing, that can sound really appealing. This is complete nonsense. And to detect biology for behavior in classroom is really scary to me. Sure. Yeah.
Brain initially was started, as you said, by long. And initially to sort of but it became more of a more focused initiative on developing the tools to do that. Yes. Because those didn't exist. Right. So now the outcome of that initiative is to develop the tools. Yes. And so far, there hasn't been so I work in pharmaceuticals, and of course, for us, the main thing that we want to see is a very robust tool to measure one specific aspect of cognition, let's just say, or even brain activity. The most established technique for brain understanding is hash type experiments for action detections. That's the hallmark that we have. Hallmark technique. What is the evolution of that today? What have we accomplished today that is a robust technique that someone like you can say, yes, I can draw that technique and draw a conclusion that is rooted in neuromechanics and also neurobiology, and then be extracted to cognitive neuroscience.
So the question is about kind of applying new tools for measures in pharmaceuticals, or kind of translational. How do we take this big initiative, this excitement and groundswell financial support, and apply it at the bench for treatment?
$250 million is not critical enough.
Yes. Okay, so 2 points. First, the Brain Initiative first round of grants were just released like a couple of months ago.
Yeah.
So the fact that we don't have big advances in a couple of months is not surprising to me at all. And $250 million is a lot of money, but it's not very wisely sent out. I think there was like 12 grants or something. Right. First wave of grants. And in terms of translational research, I think Optogenetics, for example, it's not brand new, but it's very new. It has a lot of potential for doing the same things that PatchConnect kind of analysis can do, very robust targeting and examination of activity of individual neurons. I think it has the potential to go far beyond what you can do with PatchConnect. Not only can you monitor the activity of these neurons, but you can affect that activity in very precise ways and living animals less invasively than you can do with PatchConnecting. In terms of kind of bigger things, it's always hard to say. I have a friend who is a particle physicist, and he was doing medical imaging at Memorial Sloan Kettering when I started at Rockefeller. So we would have lunch because we were right next to each other, and he would say, like, "What is the translational benefit of all of this brain imaging in humans? What can we do with this?" And the answer to that is hard to say because we're still building the tools. In addition to kind of the change in technology, I think there has to be a change in our consideration of what we're measuring. So the National Institutes of Mental Health recently announced that they're moving away from definitions of mental illness based on symptoms, so the DSM kind of definitions that we're all anecdotally familiar with. Depression is if you have 7 of these 10 things over this amount of time, to investigating kind of neuroscience. So depression is these regions are affected in this particular way. This is called depression. We're still away from both of those sides coming together and some understanding of these efforts to create better tools to do wiring. Like fMRI is great for spatial resolution, but it could be a lot better, and they're building better machines to look at individual parts of the thing. Individual parts of the face area. So there is a disruption in autism that presents there. We'll be able to see it in much better detail. But we have to rethink what we consider autism too, because we might be calling different biological things that happen to look the same symptom as the same things because we have no way of. So in terms of kind of translational things, I think we're kind of a ways away from that. Optogenetics has been theorized to be applied to humans. This closed loop optogenetic stuff, you have a little computer attached and that monitors activity, feeds back and changes that activity. The guy who invented that is an MD PhD. He's a psychiatrist, these patients invented that and was thinking about that as a way to treatment. But we're kind of far away from technology understanding. So it's a lot of words to say, like we're kind of just far away. The shift away from building models to inventing technology to build models, I think is a good thing because the problem with the Human Brain Project, the European version, was that they don't have the tools to make a thing. You need that first, and if that costs $250 million, the economic impact of depression is several orders of magnitude times that. But we have to get that's my slides. I have my information up there. This is just a plug. I just started a podcast where I talk to science people, some of them at Rockefeller, about everything besides their research. So if you want to hear about neuroscience-y stuff, don't listen. But if you want to hear about what it's like to be a neuroscientist, you can listen. So anybody have any questions after all of that? And if you're watching on YouTube, you can tweet questions at me, I guess, and I'll answer them in 140 characters or less.
I have a sort of project pitch for you. So let's say that, as far as ethics are concerned, this person volunteered, and if they say stop the experiment, you stop the experiment.
Yes.
Is that second of all, let's say that we've at some point in the future invented an MRI machine that will not have significant effect on your neural activity if you're under quantum exposure to it. I put together, not exactly a research paper, but a feasible innovation type, if you will, out of a VR suit that stimulates all your senses and also allows you to move physically. Essentially, let you be in this space and be convinced that you're in this space because you have all your regular expression and sensory input. What I'm wondering is, what if, say, we did that, so we have somebody be in that suit and also have use that as a proxy to another body that allows them to relay information from reality back to the information that's realistic and with low latency that's also safe for future as at that distance as possible. How do you think we would be able to better research the brain? Because one of the biggest impacts that we're having is we're not able to see the brain in its real environment. This is why people are coming up with these headsets. Well, I can walk around in neuroscience. You can't. Well, what if you actually could?
Flying a jet.
And how do you think that could like actually be put into? Because like it's feasible. What I've designed is feasible. The other stuff, it's not been made yet, but it can be made, and I'm all for it. Just laying the groundwork to make it.
So the question is about investigating the brain in kind of more naturalistic settings through virtual reality and a suit that kind of stimulates senses.
Slingshot reality.
Yeah, okay. Slingshot reality. And some of that is happening actually. Instead of looking at, here's a tiny step which is happening. Instead of looking at a static face, looking at a movie of a face making expression, right? And trying to get to the brain, how it interacts with the world in kind of regular life. The problem with that is analysis of that data gets infinitely more complex the more variables you introduce. So the reason we show simple stimuli in fMRI is not just because we're interested in these tiny little problems, but also because it's very controlled, and when you're looking at brain activity, you have to control for a lot of visual information. Anybody's focus can be anywhere. Anybody's perception can be anything. I had to, when I was showing people faces, I had to equalize between a bunch of faces all kinds of physical characteristics. So I ended up having to show just Caucasian faces because that contrast difference, if you're looking at different ethnic groups, can look different visually, and it can have different effects. And so something like that, it's a very interesting technical question. It's a very interesting question. Lloyd?
I think of it as far as like all the variables go, is that really what we're doing is sorting for. So if you think about it, let's go back to say the 30s. We were starting to investigate radio. Now we have, we weren't able to, with that technology, filter out one radio signal, because now we have all over us. Each one of us is carrying a radio, which is emitting on at least 3 different bands. You've got your Wi-Fi, you've got your Bluetooth, you got NFC, all of those things at once. Basically what I'm trying to say is it's really just a signal we're looking for. Given enough time, we would be able to use this in a context where we could find multiple signals.
So that's a different question. So the question is about sorting through data. You're talking about big data questions at that point. You're talking about, you have to get a lot of people in a lot of scanners and get a lot of data. I was just reading, I think before the talk, one of the, not inventors of the connectome, but one of the people who kind of introduced this idea of the human connectome was just talking about the fact that we're not in, big data is not coming down the pipe. We're here. It's what we're dealing with now. It's this idea we have more data than we can sort through. Basically we're at the point where we collect a bunch of data and then we interrogate the data instead of thinking about a hypothesis and then collect the data. The problem is, the technical constraints on what you're talking about are such that it would be very.
These things.
But also the analysis would have to be invented.
Neuroscience is invented.
It's true. It's a very big technical problem, but it's also a problem of once you have all that data, what do you do with it? We have to figure out how to ask questions at that point, which neuroscience is not really based on that right now. I scan people's brains. That's my brain. I scanned it because I had a hypothesis. You don't collect a bunch of data, brains, and then look at them because it's cost me $500 to get that image.
One question that is answered before is how the information that we receive maps throughout these pathways. We can get to the visual aspect of it. So where it goes beyond that.
So we're doing that research in animals. And say what you will about animal research, and a very big, very important neuroscience lab that uses monkeys to do visual research like that just was shut down because of the concerns about animal research. it just, we're trying, but when we think of seeing something, we think this is a simple process, right? We open our eyes, visual information. It is a terrifyingly complex computation. And even in model systems that are less terrifyingly complex, it's a terrifyingly complex system. Like reducing hugely complex thing to something slightly less complex is still, we're getting there and we're trying, but it's hard.
All right, why don't we wrap that up?
Thank you, everybody.
Yeah,
Yeah,
if you want to talk a little bit more.
yeah, I'm here.
Oh, yeah, I wish I'd done it before that. But, yeah, you should have jumped up and said something. Sorry. Next week, we have another seminar that's going to be very interesting, and it's about perception and magic and neuroscience. It's going to be on Friday the 22nd at 7 o'clock, and we're going to attempt to stream that one as well. And we'll be around. Okay. All right.
Not to piggyback on that, but my first time at Society for Neuroscience, which is a conference with like 40,000 neuroscientists, the keynote speakers were magicians showing up in neuroscience of magic.
Okay.